A lot of hay has been made about CRISPR-Cas9, the gene editing technique so precise, so versatile, so easy. It's opened the possibility of chopping and swapping DNA around like a molecular level Dr. Frankenstein. The implications are huge for humans and our genome. Like, we could alter the DNA of embryos, or, more likely, treat genetic maladies in those already born. Now, in 2018, clinical trials on humans are scheduled to start for the first time. But just as they're getting underway, there may be a problem thanks to our immune system. To understand why our immune system is being such a jerk and preventing us from treating disorders like sickle cell anemia, it helps to step back and remember exactly what CRISPR-Cas9 is. It's actually an immune response bacteria use to protect themselves from viruses. Cas9 is a protein the bacteria make that acts as molecular scissors, cutting virus DNA to ribbons. And two of the most commonly used types of Cas9 come from bacteria that can cause staph infections or strep throat. These are bacteria we want our immune systems to have a defense against. And to defend against them, our immune system has to be able to recognize the bacteria and the various proteins they make, so they can track them down and destroy them. Sorry, I called you a jerk earlier, immune system. You're just doing what comes naturally to you. To see if these two versions of Cas9 were on our immune system's hit list, researchers tested the blood of 34 donors for antibodies. They found 65% had a defense against the strep version, while 79% fought the staph variety. In another experiment with 13 blood donors, 6 of them had T cells that attack staph's Cas9. The findings are yet to be peer-reviewed, but one of the authors is the scientific founder of a leading CRISPR human therapy company, so he's got some credibility. And if our immune systems respond to CRISPR, it could mean more than just rendering the therapy ineffective. It could make our immune systems go haywire and kill us, which has happened in a previous attempt at human genetic therapy. The next question is this the end of the line for CRISPR? Good game, well played, but our bodies are just too vigilant for this revolutionary gene editing? No. We're gonna science our way around this conundrum. There are already a few solutions to this possible problem, like modifying Cas9 into a form our immune system can't recognize, or using a different protein from the oodles of other bacteria that have their own versions of Cas9. Or, if the protein gets attacked when it's in our bodies, then perhaps we could put it to work outside our bodies, modifying extracted cells before returning them to the body. Or Cas9 could be used in places where the immune system is locked out, like inside our eyes, where it could treat hereditary blindness. CRISPR has way too much potential to let a little thing like our bodies stop progress. The findings of these recent experiments are more of a speed bump than a brick wall. Medical advances occasionally have to overcome biological defenses. For example, our blood vessels in our brains make it really hard to deliver medicine to our old noodle. To learn why the blood-brain barrier is overall a good thing, check out this video here. We could talk CRISPR all day here at Seeker, so if you have any questions about it, go ahead and drop them in the comments. Be sure to subscribe, and thanks for watching Seeker.